bacteria, we want to kill it. The white blood cell puts it in the endosome, goes into the lysosome, bacterium is destroyed there. That's the idea. Okay, and then there are vesicles that serve to secrete stuff. And here's our friend the peroxisome. You see, involved in the breakdown of hydrogen peroxide, H2O2 to oxygen. How are we doing? Good, huh? Alright. So let's review very quickly the organelles of the eukaryotic cell. I just said it. So which organelle do we have? What's the main function? What are the main components? The cytosol is the solution in the cell. It holds a lot of proteins, enzymes, it holds RNA that's used to form those proteins. And also the ribosomes are in there. A lot is going on in the cytosol. It's considered to be the main compartment of most cells. However, if we think of plant cells, in reality, in most cells in a plant, the volume of the vacuole is huge. The vacuole is 90 to 95% of a typical plant cell. So that actually means that the volume of the cytosol, percentage-wise, is not so high in plant cells. In animal cells, it's probably the largest compartment. Okay, now we have the nucleus, it holds the genetic information. We have the DNA in the nucleus. There are also proteins that replicate the DNA, proteins that serve a function in transcription, forming RNA, and then there's some RNA in there that is on its way to having a function in the cytosol. And some RNA has a function in gene expression in the nucleus actually. Okay, then we have our friend the mitochondrion. One mitochondrion, two mitochondria. It's involved in ATP production and respiration. There's a couple of other essential biochemical pathways happening in the mitochondrion, but this is the main one. So the majority of the ATP that we make in our cells is made in our mitochondria. The structure, there are two membranes. There are proteins inside. The mitochondria have a little bit of DNA. There are a couple of genes in there that encodes for this. A couple of proteins. It's some 20 to 200 depending on which organism we're in. So mitochondria are a little bit special. They have a low level of autonomy within the cell. Okay, then we have our friends the peroxisome. Special breakdown reactions like long fatty acids and some other molecules. And those breakdown reactions are often characterized by the production of hydrogen peroxide, which is turned over by the enzyme catalase. That is the really sort of a marker enzyme of the peroxisome. The structure, it has one membrane, and there are specialized enzymes, remember that. The peroxisome is a relatively simple organelle. At another level, it's one of the most poorly understood organelles in terms of how it is formed and where it comes from in an evolutionary sense. We'll touch on it later. Okay, now we have the plasma membrane, also known as the cell membrane. Hey, that surrounds the cell. What is its function? It is a selective barrier for chemicals. It's not a mechanical barrier, it's a barrier for chemicals. So what I mean is not a mechanical barrier. If we push on the cell, the plasma membrane will burst or it will bend, it will give. But chemicals, molecules dissolved in water, can only cross the cell membrane, the plasma membrane, through specific transport proteins or actually through controlled processes in general. That's even true for water itself to some extent. So it's a selective chemical barrier, but it also serves to interact with the environment. The plasma membrane is what allows the interior of the cell to communicate with the outside. What is it made up of? Of lipids. When we speak of animal cells, those are mainly phospholipids and a lot of proteins. We'll get to the lipids and the proteins of membranes later. I'm quite big on membranes, so you'll learn a lot about membranes in this class. Now we have the endoplasmic reticulum, or the ER. it serves functions in the synthesis of secondary proteins. And by secondary proteins, I mean proteins that later have a function in either the Golgi, or the vacuole, or the lysosome, or endoplasmic membrane, or secreted. Secreted hormones, for instance, are all produced in the ER. And digestive enzymes that enter your digestive tract, they are produced in the ER originally and then secreted from the cell. Yeah. So, secondary proteins. The ER is also largely responsible for the synthesis of lipids and phospholipids that make up other membrane systems of the cell, except for the chloroplast. That's the communication between the chloroplast and the ER in plant cells. Serves a function in detoxification, and that tends to be a special area of the ER that we call the smooth ER. So I always like to say that people who smoke a lot and probably in their lung cells have some expanded surface of smooth ER. There's a special enzyme called cytochrome P450, which has a function in detoxification of very hydrophobic molecules. That adds oxygen, makes it more hydrophilic, and it can be degraded. There here. Yeah, and the ER, we can also say, forms the nuclear envelope that is the barrier around the nucleus. Okay, then you have the Golgi apparatus, thanks to Mr. Golgi. What does it do? It takes proteins and lipids from the ER, modifies them, and you can see this as a post office shipping center, from where secondary proteins are sorted to go to their correct destination. Okay, now we have the lysosome in animal cells. The function is mainly turnover, degradation. So the environment is favoring hydrolytic reactions. That's a low pH environment, it's acidic. In plants and in yeast, we call the same organelle the vacuole. It has more storage function there. Okay. Now there are several types of vesicles that connect all of these compartments. So all of these compartments are related. The membrane lipids components travel between the different compartments by means of vesicles. Therefore, we call this one system the endomembrane system. I spent 4 years working with the endomembrane system in yeast. How are we doing? Did it all repeat or be good to know again? Alright. Now, other organelles that are not directly related to the endomembrane system. In plants, we have our friends the chloroplast, function in photosynthesis. In a chloroplast there are 3 membranes. There are tons of proteins in there. The chloroplast also has its own DNA, it has RNA, so it is also a little bit autonomous. In fact, the chloroplast is a little bit more autonomous than the mitochondrial. There are more genes, I think, in the typical chloroplast genome than in the mitochondrial genome. Okay, plant cell or fungal cell is surrounded by a cell wall. The main function is support to give the cell its shape. It's a mechanical barrier, and in plants, its main component is cellulose, and then hemicellulose and other components. There are a couple of them. Cellulose is probably the most abundant polymer on our planet. Cellulose is composed of this glucose, 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 the polymer of glucose. All eukaryotic cells have a cytoplasm, which is the cytosol on the organelles, said that. Again, then we can add it all together. We can say the cell is the cytoplasm and the nucleus and the plasma membrane. And then if you're a fungal cell or a plant cell, then you even add in the cell wall. All right. Okay. That's a summary of the structure of the eukaryotic cell. We will look at much more detail later at the mitochondria, at the chloroplasts, and especially at the ER and the secondary cavity. Are we good? Okay. Now let's think about the chemical composition of a cell. I didn't take this picture from the present book, it doesn't matter. You need to have something. So here we have an EM picture, thin section of bacterial cell, and if we would just imagine that we would blow this up and color this a little bit, what we see is we see some nucleic acid molecules in here, probably DNA. We see a lot of proteins, and there we see a couple of membranes. But what's really chemically all the stuff in there? Well, the majority is really water. So water is 70% in the average cell. In plant cells it tends to be more because of that natural. So plant cells, the water content can go up to 99%. If we're thinking about fat cells, cells that store fats in our body, and maybe the water content is lower, maybe around 30%, I don't know. On average, roughly 70%, give or take 25%, I like to say it's 25%. So a lot of water. Now, let's do something fun. How does it work? We can do something here. I can say this. Alright. And now, I have, okay, I think I have to go here. Come on. Maybe it will work actually. Let's see. It works. Okay. Alright, so I think a lot of students don't understand the numbers that are involved. If I say that 70% water, it's just fun to think of how many water molecules there actually could be in a cell. Interesting. I like numbers, okay? So, now, average size of a cell is something like 10 micrometer. That's roughly a meter. Right? A micrometer is 100,000 times less than this distance. Okay, and that's roughly the average size of a eukaryotic cell. Bacterial cell is smaller. So if we roughly estimate the volume, if we say that cell is a cube, then we have, so this is 10 to the negative 5 meter, right? So then the volume, I take it to the third. The volume, therefore, of our average cell is something like 10 to the negative 15 meter to the third cubed. Am I good? I'm very nervous about math in public. Bear with me. So, meter cubed is of course 1,000 times more than a liter. So that means that we have 10 to the negative 12 liters. Now there are 55 moles of water in a liter. Yeah? So we have 10 to the negative 12 liter times 55 moles of water times Avogadro's number, 6 times 10 to the 23rd. I did this math, and we get to, I can't read that. We get to, yeah. Okay, I did this math and it was roughly 3 times 10 to the 14th molecules of water per cell. I want you to understand this is a huge number. Yeah? There are 6 times 10 to the 9th people on the planet. And there are roughly 100,000 times more water molecules in a small cell than that there are people on the planet. So you have to understand the numbers are huge. Okay. Now, also in cells we have things like organic molecules and we have inorganic ions. And the main ion is potassium. And potassium will be present, at roughly, 100 millimolar. So that is something like 500 times less in concentration than water. So we have, if we think about potassium, we would have something like 1.5 times 10 to the what is it? 12 potassium ions. Huge numbers. The potassium becomes important later in the class when we talk about the function of neurons. If you do the same type of math and you think about proteins, there's about a 10% weight by volume protein content in the cell. If you take for the average cell, they say roughly 10,000 different proteins in the cell. If you do the math, and I did it, trust me, the average protein has 100,000 to 1,000,000 copies in the cell, and then there are 10,000 different constituents. What I'm telling you, there's a lot in that small cell. Okay? All right. Now, I've made it messy. I don't know if I can. Okay. I don't know if I can. Okay, so we have a lot of water, right? And we have inorganic ions. Potassium is the dominant one. The name potassium comes from that, actually. Potash is what you have left in the pot after you burn wood. You burn wood in a pot, you have ash left. That's called potash. The main component is potassium. Why? Because it's the main inorganic component of life. The organic components all burn, react with oxygen, leave as a gas. And like fireplaces, etc. And remove a lot of potash. It's a Dutch word for it. Okay, another important ion is magnesium, which is the second most abundant one. Calcium and sodium are present in lower amounts inside cells. But also some phosphate, etc. Together that's roughly 1% of maybe a bacterial cell. Then we have other organic molecules like nucleotides, the subunits of DNA and RNA, amino acids, sugars, fatty acids. That's another couple of percent together. Okay? And then we have the macromolecules. Together that's about a quarter of the weight of the cell. And the macromolecules is together the DNA, the RNA, the proteins. And the proteins are the largest fraction of the mass, of especially the interior of the cell, and then polysaccharides. Now if you speak about plant cells, because of the presence of the cell wall and the presence of starch, the polysaccharides can be a larger fraction of those cells. If you take the average cell, proteins is the main macromolecular fraction. Okay, and then we have lipids forming the membranes, and lipids in the form of triglycerides that are the fats for storage. Okay. Really good still. Let's look at our macromolecules because they're so important to the functions of cells. So, here's a little bit of review. I'll go pretty rapidly. DNA serves as genetic information carrier. The subunits are 4 different deoxyribonucleotides. And we have RNA, ribonucleic acids, function in general protein synthesis for ribonucleotides. We have the proteins. Many different functions exist for proteins. Proteins give the cells their real functions and their structures. A very important class of proteins are the enzymes, but there are many, many more, like structural proteins, or motors, or receptors, or transporters, on and on. Subunits with 20 different amino acids. And we have the polysaccharides. They have functions in support, like cellulose, or storage for energy. Think of glycogen in our muscles, polymer of glucose, or starch, also polymer of glucose in our muscles. Subunits, sugars. Okay, carbohydrates, you can say. Then we have lipids. Functions of lipids, they form the membranes, and that's for phospholipids and related molecules. And for energy storage, you have the fats, the triglycerides. Okay, subunits are fatty acids and glycerol. Okay, what about how macromolecules are formed? I think it is useful to summarize that there is actually sort of a general law for the synthesis of macromolecules. And most textbooks don't do this. But actually, when you think about how a macromolecule is formed, there's a general principle and it's always the same. What are macromolecules? Big molecules. They're polymers of subunits. There always are subunits that were pre-formed. They are connected by chemical bonds, or covalent bonds, or strong bonds. The breakage of the chemical bond between the subunits to form the polymer releases energy. Therefore, the formation of the polymer from the subunits is something that requires energy. So it takes always energy to form a polymer from the subunits. Therefore, there is no case where polymer formation is spontaneous. Therefore, cells control the formation of polymers by means of their enzymes, and they do this with the input of metabolic energy, especially using ATP. We'll see that later. So what is the general principle? The general principle is that the subunits are individually pre-activated. And what that means is there's some chemical group added to the subunit that makes it pre-activated. So the inclusion into the polymer becomes a downhill, spontaneous, favored reaction. Ultimately, the energy to activate the subunits always comes from the subunits of RNA, that is, the ribonucleotides. And therefore, it is true that ribonucleotides, the subunits of RNA, have a key role in the control of all of the formation of macromolecules, and therefore the formation of the entire cell and all of metabolism. This is one reason why many scientists, including me, think that it is likely that early in the evolution of cells, that actually a lot of processes were controlled by RNA and its subunits. We'll get back to that. Now, the most important ribonucleotide that serves as the ultimate energy source for the activation of our subunits to form macromolecules is ATP. Okay, so here we have the process in the picture. Let's say this is a polymer. Okay? Let's say these are subunits, and there we have the covalent bond that keeps the subunits together. Just told you this is a bond that stores energy because when we break it, we release energy. How can that be done? In the presence of water, in a hydrolysis reaction, as it's called, we can break this one subunit of the polymer, and we see the release of a little bit of heat. It happens for all polymers, always true. So such a breakdown reaction, because it requires the consumption of water, is called a hydrolysis reaction. Hydro, or water, lysis, hydrolysis, or breakdown. Okay? So a hydrolysis reaction is a breakdown of a polymer, where water is consumed and energy is released. We break down the covalent bond between the subunits. We recover the subunits. This is where the peptides that we have here now had a tripeptide, and we had a dipeptide, and a single amino acid. Okay? In a reverse reaction, in the formation of polymers, which we call synthesis or biosynthesis, or condensation, we have the following. We had a subunit that was pre-activated. It's bound to some other group, it depends on which type of polymer we're forming, and that chemical bond here, this chemical bond here, makes it possible that the subsequent addition of our subunits to the growing polymer is a downhill reaction. It's therefore a favored reaction, and it has to be favored. Okay? So, that then means that in this formation, from the activated subunit, we again get a release of a little bit of heat after the reaction. And that's how the polymer grows. Now, if we ignore this activation of the subunit, because what happens actually is that this activating unit is recycled all the time, and in the overall reaction, if we just think about how the subunit is first activated and then added, in the overall reaction, water tends to be formed. Therefore, we call these types of reactions condensation reactions. The overall formation of polymers are condensation reactions because there's a net consumption of water. In a reverse, in the breakdown, there's a net release of water, hydrolysis. Net consumption of water here, net release of water in the synthesis. She would say. I think I found the wrong box. I'm going to say it again correctly. In the synthesis, the net reaction, water is formed, we call it condensation reactions. The breakdown in the net reaction, water is consumed, we call it hydrolysis. Did I say it right? Alright. Okay, so there we have our water that's consumed there. Okay, formed. It's time to start, that's clear. So, please take a moment to pick up your take-home exam. You need to have an answer sheet also. Treat your answer sheets nicely so they will scan well. I'll get these back from you next week.